that can lead to regulation of a number of downstream cellular functions, such as ion channel regulation, secretion, and NFAT activation. And many people, a lot of people in this audience have been working on these mechanisms for many years now. So why did we get interested in this project? Was the fact that, the tissue at that time that we were interested in was salivary glands. Today, I'm not going to talk about that. Salivary glands sense a variety of different agonists at different concentrations during the activation process, which leads to fluid secretion. So the question is, how do the cells actually respond to these sort of graded agonists stimulation? At which end of this stimulation curve do they work? And how are the functions regulated at either low end of an agonist or a high end of an agonist? So as I just mentioned to you, the depletion of ER calcium is very important for triggering the whole SOCE process. And you can easily imagine that at very low agonist concentration, ER calcium depletion will be quite low. So calcium concentration will be pretty high. And reversely, at high agonist concentration, you'll have very low ER concentrations. And cellular responses increase as agonist concentration increases. So when you look at the agonist, the calcium signals in the cytosol, people have identified oscillatory calcium signals or very sustained calcium signals. And typically, these oscillatory calcium signals are generated by low-end stimulus intensities, whereas these sort of more sustained calcium signals are generated by high-end stimulus intensities. So how does the SOCE now work under these conditions, right? And why do we need differential sensing of the ER calcium concentration? So this is where now the Stim proteins come into play. And at least that's our concept of how they work. So Stim2, which has a lower calcium affinity, can respond to small decreases in ER calcium. Stim1 with a higher calcium affinity requires a higher and more substantial depletion of ER calcium. So is that actually the case? So if you look under resting calcium conditions, Stim1 is pretty well tracking, and this is overexpressed protein. It's only after stimulation you see clusters. Whereas in the case of Stim2, they are clustered even under resting conditions. And when you stimulate the cells, you'll see not too much of increase in the number of clusters, but an overall increase in the intensity of these clusters. So what got our immediate attention is this pre-clustering of Stim2. And we wanted to find out what's the relevance of this and what determines those pre-clusters. So at that time, when Wheeling published this paper in 2015, she looked to see the responses of these two Stim proteins at different levels of agonist. So she used the 1 micromolar as a low agonist concentration and 100 as a relatively high agonist concentration. So under basal conditions, Stim2 is already clustered. The clusters increase at low agonist and don't change a whole lot more beyond that at a high agonist. Now we know that there is a change in intensity between a 1 and 100 micromolar. On the other hand, if you look at Stim1, we hardly see any response at the low agonist concentration, responds very robustly at the high agonist concentration. If you co-express ORI, it does not rescue this low agonist response, but you do get very nice responses at a high agonist concentration. Now, since cells have all the three proteins together, what happens if you express Stim1 and Stim2? Well, in that case, we find that the Stim1 also co-clusters with Stim2. So Stim2, in the sense, endows the Stim1 the ability to cluster at these low agonist concentrations. And when you have the three proteins together, you see with very nice co-clustering of all of them at 1, and then further clustering at 100. So what do they do? So the data that we published earlier suggests that Stim2 increases the agonist sensitivity of the SOCE activation. So if you plot a curve like this, where you have the calcium increase on the y-axis and calcium concentration here, this black line is the control cells. So this is your endogenous SOCE. If you knock down the Stim2, you get this nice right shift in the curve, suggesting a decreased sensitivity of the cells to recognize and be activated by the agonist. Now, what is the role of STIM2 and STIM1 under these conditions? So if you knock individually these two proteins down, you realize that the STIM1 is absolutely important to activate ORI. In the absence of STIM1, you completely lose this oscillatory calcium signal at the lower end and even the sustained calcium signal at high agonist. But if you knock down the STIM2, all you do is you decrease the frequency of these oscillations, and you might convert part of the cellular response to the high agonist, and some cells will start showing oscillatory patterns even at 100 micromolar carbachol. So then the question was, if STIM2 actually, STIM1 is the Stim protein that is gating ORI, and it cannot respond very well at low agonist, how is this whole thing working, right? So then when Krishna was in my lab, he looked to see what is this role of basal pre-clustered STIM2, what exactly does it do? And one of the suggested roles for STIM2, and this has been reported by Tobias and other people, is that it regulates basal calcium entry and serves like a housekeeping function. So when he overexpressed STIM2 and co-expressed ORI with it, he in fact saw co-clustering between these two proteins. And under those conditions, he could actually increase the calcium influx more than what you get with expressing STIM2 alone. So when you express STIM2 in cells, you actually get this increase in basal calcium influx. And that is mediated through its recruitment of endogenous ORI. So our hypothesis was that if STIM2 is able to recruit ORI, then it has to be in sort of a partially activated conformation, even in the resting conditions. And Krishna actually described that, and he did further experiments to actually establish that. And this is already published. But the take-home message from that paper was STIM2 can recruit and activate endogenous ORI in the absence of stimulation, suggesting it is likely to be in an activated state. And in an activated state, in extended or whatever conformation you want to call it, STIM2 can interact with and activate STIM1 to cause STIM1 ORI activation and calcium influx. So we got very interested in this problem of what controls STIM2 pre-clustering. And we didn't want to again continue using overexpressed proteins because maybe that itself creates an artifact. So when Moaz joined the lab, he came in with this idea of trying to knock in a tag into the endogenous STIM2. And this paper just got published, and I have discussed this briefly at the ECS meeting as well. So he established this knock-in cell line that expresses endogenously STIM2 that is tagged with a venous. So when you look by confocal microscopy at the ER, the STIM2 very nicely decorates the ER network along with the ER marker. If you now do a turf experiment, you can see ER at the subplasma membrane level, and the STIM2 is very nicely punctate in this ER that is situated right below the plasma membrane. These are just enlarged pictures of the same. So is this a normal STIM2? Can it still respond? Yes, it can still respond. So here are the responses to carbachol, low carbachol, and high carbachol. And we have now calculated the number of STIM2 clusters as well as the intensity. And here is a video showing what does this endogenous STIM2 look like. So what is immediately apparent is that they are pre-clustered. A lot of the clusters are highly mobile. And if you remember my picture with the video with the expressed STIM2 protein, a lot of those clusters were very, very stable with the overexpressed STIM2. This is endogenous STIM2. It seems to be highly mobile, or in the sense it's flickering quite a bit. However, when we look very closely, there are a number of these clusters which are pretty stable. So we would like to know what exactly these stable clusters are doing and what's the difference between them and the mobile clusters. So to do that, actually, Moaz used the trick that have been used by other people and to determine and try to quantitate these stable clusters and to determine their pattern or how they change after stimulation. So what he used is he did this trick where he overlaid two successive frames of the video. So, for example, here it's 0 minute and 1 minute. So they're 1 minute apart. And if you overlay them, and they're pseudo colored, the white clusters then denote the clusters that are superimposed and they're not changing. So we call these stable clusters. They're relatively stable. We don't know that they will last forever after stimulation, but at least for a 1 minute, 2 minute, 4 minute time frame, we can see them. So, and after stimulation, the number of stable clusters increase. Similarly, at a high carbonyl concentration. So this became really interesting that there are these stable clusters at basal, under basal conditions, resting cells, and following stimulation, these increase. So the change in mobility has also been previously reported for STIM1 and NRI. And it's been always associated with sort of an activated conformation. So we were thinking at that time that maybe these stable clusters represent what we had originally thought of as the activated state of STIM2 proteins. There are less of them because we are now dealing with endogenous protein and not the overexpressed, which tend to crowd the ERPM junctions. So we went back to confirm some of our previous observations. Does this STIM2 stable clusters play a role in recruitment of ORI? Yes, that is shown here. If you identify a stable cluster and you then trace the recruitment of ORI, here we've expressed ORI using this TK promoter, which gives you very, very low level of ORI1 expression. So it becomes very easier to see small ORI clusters. You can see now that ORI actually gets recruited to the same stable STIM2 clusters. And once recruited, it actually becomes pretty stable and it's recruited within that STIM2 cluster. This just shows you the temporal course of that recruitment. Similarly, for STIM1, which is tracking initially, it finally gets recruited into these clusters, which are the stable STIM2 clusters. So that is very nice because it shows that these stable clusters have a purpose. Upon agonist stimulation, they're able to recruit STIM1 as well as STIM2. So this might actually form the SOCE microdomain. So what determines these pre-clusters? How is this STIM2 there? Because it seems to be playing such an important role in the recruitment of STIM1 and ORI. So we look to see whether they are associated with the mapper decorated domains. And yes, STIM2 co-localizes very well with mappers. In fact, there's more STIM2 injunctions following a mapper expression, and we could talk about that later. So the endogenous junctions, preformed ERPM junctions, have been reported to require ECT2 and 3, whereas the junctions that are formed after stimulation are calcium dependent, and they have been suggested to be require ECT1. We didn't look at the ECT1 junctions. We were more interested in these preformed junctions. So when we knock out the ECT2 and 3 in these cells, we actually see a loss of this basal STIM2 clustering. And when we looked at the ER and the mapper in these conditions, this was not much of a surprise because looks like the ER plasma membrane scaffolding also requires ECT2 and 3, as do the junctions as seen here with the loss of the mapper decoration at the turf plane. So are these just preformed junctions sufficient? So if you have a junction there, can STIM2 get to that junction? Or are there other cellular cues that are guiding this STIM2 recruitment into the junctions? So based on Colin Taylor had published a paper a while ago talking about this IP3 receptors that are sitting very close to the plasma membrane, and they can sense the IP3 that is generated and locally within that domain, and they are able to respond. So we were very interested to see whether these IP3 receptors that are sitting very close to the plasma membrane might have a role in activation of STIM2. So what Moaz did was he looked at the localization of IP3 receptors in relation to STIM2 protein. And you can see that they are very closely, in close proximity, you see these clusters. Although the IP3 receptor clustering under these conditions is not very clearly clustered, this is an expressed IP3 receptor type 1. And these two puncta, they are partially overlap, but they're not completely overlaid. Now, is this IP3-1 necessary? So here we looked at the effect of knocking down the IP3 receptors, and we actually saw that the Stim2 pre-clustering is completely gone. And when you overlay the protein, you see that the IP3 receptors are quite nice, and they lose this Stim2 overlay that you see when both proteins are present in the cell. I think this might be a... Oh, sorry, I misspoke. So here we are using an ER marker, and we are knocking down the IP3 receptors in the cell. So this is actually ER. So the ER localization is not affected. Only the, only the Stim2 localization is affected. I apologize for that. So to confirm this, we did the same kind of experiments now with the triple knockout cells which lack IP3 receptors. And again, we see that the basal clustering of Stim2 is gone. Is this functional requirement for IP3 receptors? Yes, because if we use a gating deficient mutant of IP3 receptors, we don't see the clustering. However, if we put the R1 IP3 type 1 back into the cells, we can recover the clustering. So this pre-clustering of Stim2 is absolutely dependent on the presence of functional IP3 receptors. So what exactly are they doing? If it's functional, then it has to be related to the decrease locally of calcium within that ER calcium pool. So to do that, what Moaz did was he used this ER large echo, which is a ER calcium sensor dye. And the way we did this experiment was we stimulated the cells with phoscolin, which actually increases the IP3 receptor function in the presence of the existing levels of IP3. So we didn't come in with an agonist in this case. We're just using phoscolin, which enhances through pKa phosphorylation of the IP3 receptors. So we first identified an increase in the Stim2 clustering. And you can see here, this is the stable Stim2 clusters. You see an increase. And based on our predictions, locally associated with these Stim2 clusters, we should see a decrease in the ER calcium concentration. And in fact, that's what we saw, and that's shown here by this red graph. And this is just the time course of this store depletion after addition of phoscolin. You see that the Stim2 signal very nicely increases, whereas the ER calcium concentration, that fluorescent signal of large echo decreases with time. In the triple knockout cells, we don't see that decrease. And just to make sure that this is in fact true, Moaz tried both his knock-in cells as well as a wild-type HEK cells, and they both behave the same. So how is the function maintained under these conditions? Like I told you, there has to be a certain ambient level of IP3 in the cells. Can we actually block PLC to decrease that ambient level and see whether then this clustering gets affected? And in fact, that's what we saw, that there is a requirement for this sort of basal PLC activity to achieve this basal clustering. So when we treat cells with this PLC inhibitor, you lose the basal clustering. So in resting cells, the Stim2 pre-clustering is determined by IP3 receptor function, that is through basal PLC activity. And so is this, the sensing of Stim2, is this really involved in this whole process? So we got these really neat constructs from Mo, where he has taken the Stim2 and replaced that with a Stim1 N terminus. So here, this is basically Stim2 with a very high affinity EF hand domain at its N terminus. So we would have expected that there will be a decreased response and it would start behaving more like Stim1. And in fact, that's exactly what happened. So if you look at now the wild type STIM2, you get these very nice stable clusters. Whereas when you go to this chimeric STIM2 construct, you see that there is no stability in that. And this is just a movie where you can see the pattern of this wild type TK-STIM2 construct as well as this chimeric TK-STIM2 construct. So there is actually, we are losing completely the stable STIM2 clustering just by affecting the N-terminus. So this is actually a very important observation that we made. So it's suggesting that, this pre-clustering is not a random event. It's actually a result of the sensing of local ER calcium, which then depends on the IP3 receptor function. So does this have any function? So going back to its suggested role in basal calcium influx, we now expressed TK-ORI1 in STIM2 knock-in cells. Earlier, I showed you a cell where we had STIM2 clusters, and then ORI was recruited to the STIM2 clusters. So not all STIM2 clusters are interacting with ORI under basal conditions, but there are a few clusters. So in this case, when we overexpress the TK-ORI, you now see that several of these clusters are co-localized with ORI. So there is a certain number of STIM2 endogenous STIM2 clusters that can co-localize with ORI channels under basal conditions. And of course, this increases when you stimulate them. And it's through these basal co-clusters, we believe, that STIM2 can carry out the basal calcium influx process. So if you knock out the ORI1, you will lose this calcium influx. When you knock out STIM2, you lose the basal calcium influx. So this becomes a very important function for STIM2 under resting conditions, STIM2 preclusters. But there's also, it is very important after stimulation, especially at low level stimulatory conditions. So this is my last slide. And this is the model that Moaz had shown in this paper that just appeared, I think earlier this week or last week, where he has proposed that STIM2 is localized within this ERPM junctional ER, let's call it that way. And if the STIM2 happens to be close to a functional IP3 receptor that is sensing these inputs, be it IP3 or cyclic A, dependent PKA input, then the activity of this IP3 receptor is enhanced. That results in a local ER calcium depletion. And the STIM2 that is sitting close to this or moving somewhere very close to this IP3 receptor can sense that depletion. And then, it undergoes the usual conformational change that everybody has proposed, part of which that involves it's being scaffolded to the plasma membrane. And we believe that is what is causing the stable STIM2 clustering. And once it's in this state, it can now recruit ORI1, and probably this sort of complex, which is consisting mostly of STIM2, there may be one or two instances where STIM1 might be present as well. You get the basal store-operated calcium influx. And once you start putting the agonist on, depending on how much the store gets depleted and how much agonist goes on, you'll get clusters which have either STIM2, STIM1, or maybe STIM1 alone at these very, very high agonist concentrations. So with that, I will stop, and I hope I meet everyone in person, including these guys who work very hard in my lab every day. Thank you, everyone. Hello?
Thanks, Indu. That was great. Mo, are you there?
Should I go to end show mode, or what should I do, Martin?
We can switch straight to Moaz. Sorry, Mo, I'll let you take over. I wasn't sure if you were there.
No, it's okay. I'm here. Yeah. I mean, Moaz, can you share your slides?
Yes.
All right. The floor is yours.
Indu, can you stop presenting?
Oh, sorry.
Thanks. Thank you. Nice talk. Thank you.
Oh, somehow it is not letting me share only PowerPoint, rather it's asking to share a screen.
Go to window, Moaz. That works.
Okay, okay, got it. So got it there. In the somehow I don't know the reason. Okay.
You okay, Moaz?
Yes. Sorry.
Arrow at the bottom, Moaz.
It's,
Just click on that arrow and say.
although no, my PowerPoint is open, but it is not showing in the tab. So
It took some time for mine also
I
to kind
think
of.
you should first close if the PowerPoint is open, you should first close that, then you can share.
you mean
Don't go into presenter mode. Just first keep it in the normal. Just open your file. You remember how you did it yesterday? Or
yes, I got it.
Okay.
Can you see now?
It's coming up.
Yes. Okay, now you can go full screen. Excellent.
So it's visible now, right?
Yes.
Okay, thank you. So I would like to thank my supervisor for giving me this opportunity to speak at this Calcium Society level, and I would like to thanks for the invitation to Martin and Katja, and for the and Mohammed Tribak for the introduction. So I would like to continue from where in the stop her presentation. So basically, I'm going to discuss the functional communication between STIM2 and IP3 receptor and how it coordinates cellular response at subthreshold stimuli. So from Indu talk, we now know that STIM2 and IP3 receptor has functional communication, and once there is some activity of IP3 receptor, it lead to the clustering of STIMs or I, and which cause SOCE or calcium influx. So because IP3 receptor is playing very crucial role here. So let's have a little closer look like how this is regulated. So we know that through muscarinic receptor, it activates the phospholipase and which generate the IP3. And if IP3 bind to the IP3 receptor, it gets the channel activity. And in parallel, growing evidence suggests that through beta adenoreceptor, it activates the GS protein, which can activate adenyl cyclase, and this adenyl cyclase generates cyclic AMP, and this cyclic AMP can activate protein kinase A, and this protein kinase A can phosphorylate the IP3 receptor, and the phosphorylation of IP3 receptor basically potentiates it for its open probability of the channel in the presence of IP3. So we know that basal activity of these two pathways basically cause STIM2 clustering. So in our ongoing work, we want to investigate whether these two pathways, how these two pathways basically regulate STIM2 function, STIM2 clustering, and how it regulates SOCE and downstream functions. And David Yule and others have reported that there is a synergy between these two pathways. So we'll come to that. So what we did, we wanted to know that if we block these two pathways individually, what happened to the Stim2 clustering. Although Indu has mentioned in her talk, but I will go in detail. We aim to block this PLC using the U compound. So we use both active and inactive compound. So here is the Stim2 basal cluster, and when we use inactive compound, it does not cause any effect on the Stim2 basal cluster. But when we stimulate the cell with forskolin, basically it causes significant increase in the Stim2 clusters. And when we use the active compound, it caused substantial reduction in the Stim2 clusters. And when we treated the cells with the forskolin, it does not cause increase in the Stim2 cluster. So this suggests that here you can see with the active compound and the inactive compound, and you can have a comparison here. But what I interpret from these data set is that if you block this PLC pathway, and even if you block this PLC pathway and even you stimulate with this pathway, is not sufficient to cause some activity of the IP3 receptor dependent Stim2 clustering. So next we wanted to see what happens if we block the cyclic AMP pathway while keeping the PLC basal activity functional. So we use H89 compound. We blocked the when we treated the cell with H89 compound, it also substantially reduced the Stim2 basal clusters. And when we stimulated the cell with forskolin in the wash condition, it causes Stim2 cluster back to the at least to the level where we see in the basal condition. And you can see the quantitative analysis here. And similarly, when we used forskolin in the presence of H89, it is not able to cause Stim2 clusters back. And it looks like it looks like very similar to what we see with the inhibition with the H89. So it suggests that this pathway is very crucial under the basal condition. So we hypothesize that if we block this pathway and we stimulate this pathway maximally, then what happens? So here's the Stim2 basal clusters, and when we inhibited, when we use the H89 compound, it basically reduces the Stim2 basal clusters. And when we put the 100 micromolar carbachol in the presence of H89, still it is able to cause significant increase in the Stim2 cluster. So this suggests that at high agonist stimulating this PLC with high agonist in this scenario, cyclic AMP pathway becomes redundant. Next, we wanted to know what happens. So here is the video. Like you can see, like when we use H89 compound, and then we stimulated with forskolin with and without wash. So here you can see the Stim2 clustering is back, but not in this case. So I will move on with our finding. With these findings, basically what we conclude that under the basal condition, both cyclic AMP, both PLC are IP3 signaling pathway and the cyclic AMP pathway are very critical in determining the IP3 activity, basal activity. So David Ewell labs and others have already reported that there is a synergy between these two pathways. And so we question, like, what happens to the Stim2 clustering when we treat the cells with subthreshold stimuli, and how it regulates downstream functions. So to get the insight of synergistic involvement, what we did, we titrated down the carbachol concentration, and at 0.5 micromolar carbachol, we found that there is no increase in the Stim2 clustering, although we still analyzing at the individual cluster level, but even for long term for a treatment for several minutes, basically up to 10 minutes, you can see this is a flat line. There is no increase in the Stim2 clustering with the carbachol only condition. And similarly, we did for the phosphorylcholine, and when we titrated down into 2 micromolar concentration, there is no increase in the Stim2 clusters. And interestingly, when we started the experiment with 0.5 micromolar carbachol and we treated cell with 2 micromolar phosphorylcholine, then it causes significant increase in the Stim2 clustering. You can see here, this is like a significant increase here, although I'm not showing the stat here, but we know that this is significant increase. So next we wanted to know that based on this observation, that what happens to the ORI1 cluster and Estimon cluster under these synergistic stimulus. So basically, in the subsequent slide, I will say these two concentration of the compounds together as a synergistic stimulus. So when to test the ORI1 clusters, we express TK ORI1 in the Stim2 knock-in cells, and under the basal condition, we see that there is no ORI Stim1 cluster which is expected. Although when we used 0.5 micromolar carbachol, there is no increase in Stim2 and ORI clusters. But when we use the synergistic condition, clearly there is an increase in Stim2 clusters and also causing ORI1 cluster, and these two are perfectly co-localizing, which can be seen in the line scan data here. And when we knock down the Stim2 in the same experimental setup, you can see the Stim2 cluster, Stim2 is gone because we use SI Stim2, and in this condition, there is no clusters formation of the ORI1. So this suggests that Stim2 is playing a critical role in the synergistic condition. So next we aim to test what happens to the Stim1 clusters. So we expressed TK Stim1 in the Stim2 knock-in cell line. Under the basal condition, it tracks and it don't respond to 0.5 micromolar carbachol. But when we use the synergistic stimulus, it causing clear Stim1 clustering, but also there is a Estimon which also tracks in the cells. So it's a kind of moderate clustering of the Estimon, which can be seen in the line scan after synergistic stimulus. There is a co-cluster of Stim2 and Stim1, while under the basal condition hardly there is any cluster. Although frequently sometimes we see there is some cluster, but when we closely look at that, we found that these are basically track of Stim1 which is passing through the Stim2 clusters. So we don't know whether that is real cluster or it's just like an overlap of the intensity. So all these experiments we were doing in the Stim2 knock-in cell line. So we wanted to get further evidence that what happened to these stims and stims basically in the wild type cells. So what we did, we expressed TK Stim1, and which is tracking under the basal condition, what we see in the knock-in cell line. Don't respond with the low carbachol, but when we put the synergistic stimulus, is causing clustering exactly similar to what we see in the knock-in cells. And when we knock down the Stim2, these clustering of Stim2 is substantially reduced. And to get further insight whether these are functional Stim clusters, so as Indu presented that stable Stim2 is the functional population of the clusters. So we did stability analysis. Here you can see when we put the synergistic stimulus, there is a formation of a stable Stim1 cluster. So that represent, with this data, we interpret that there is a functional population of the Stim1 in the synergistic upon synergistic stimulus, while when Stim2 is knocked down, there is no formation of a stable Stim1 cluster. So clearly there is this suggests that there is a formation of a stable cluster. So we wanted to further know the functional relevance of these Stim2 cluster in terms of calcium entry. So we used Fura2A dye to trace the calcium release and influx. And we used phosphorylcholine only condition. There is no release of calcium from the IP3 receptor. Although when we treat the cells with 1 millimolar calcium chloride, it causing some basal level of calcium entry. And when we treated the cells with only carbachol, which is 0.5 micromolar, it causing some calcium release, but there is no such a substantial increase in the calcium influx. And interestingly, when we use the synergistic stimulus, it causing substantially increased calcium release. And the most important thing is here, we see a significant increase, and it's like several fold basically increase in the SOCE. And even more interesting that when we knock down the Stim2, and there is no effect in the Stim, there is no effect in the calcium release, but calcium influx is substantially reduced. You can see the bar graph here, and you can see the difference here, like several fold. Although you can see the calcium release with phospholine, there is no, although with carbachol only there is some calcium release, but with synergistic effect there is almost twofold change. So this suggests that a synergistic condition with synergistic stimulus in our experiment basically sufficient to drive stim ori clustering and calcium influx, substantial calcium influx. So next we wanted to know, and based on these data, we build a model using our data and data published in the literature, mainly from the Mohammed Tripak lab, Anand Parikh lab, and our lab, which suggests that cyclic AMP component are scaffolded by ACAP79, and the ORI1 our SOCE component also scaffold to the same ACAP79. And any even very small stimulus of these pathway and these cyclic AMP pathway together cause a modest increase in the IP3 receptor activity, which is sufficient to drive clustering and SOCE in this signaling microdomain. So next we wanted to know, because ACAP is playing a very central role here, that is scaffolding multiple complex to coordinate multiple inputs. So we hypothesized that if we knock down the ACAP79, it will lead to the depletion of the pKa in this signaling microdomain. And knocking down the ACAP79 in the Stim2 knock-in cell line basically causing substantial reduction in the Stim2 clusters, and there is no increase with 0.5 micromolar carbachol. But you can see in the synergistic stimulus, there is any substantial increase of the Stim2 clustering as compared to what we see in the control. But when we put the 100 micromolar carbachol, which is quite high agonist concentration, and this is able to cause a Stim2 clustering back to the what we see very close to in the control. And if you look at the video here, when we knock down the ACAP, pre-clusters are gone, and whatever the leftover cluster basically are very mobile. And when we use synergistic condition not causing Stim2 clustering, and when finally we used 100 micromolar carbachol on the top of the ongoing experiment, which causes Stim2 clustering back to which causing significant increase in the Stim2 clustering, although it seems less than what we see in the Stim2 knock-in cells as a control, but we are still analyzing more and more cells to make any statistical comparison. So next we wanted to know that what happened to the IP3 receptor in the same ACAP79 knockdown condition. So basically, we found that there is no such any substantial effect on the IP3 receptor 1 localization in this condition. And we also wanted to test what happened to the calcium release and calcium influx. So here is the control experiment. You can see a synergistic stimulus mediated calcium release and influx. But when we knock down the ACAP, the same synergistic stimulus causing a substantial reduction in the calcium release as well as influx. So this suggests that ACAP79 mediated pKa function is affected, because affected because if its pKa is depleted in this signaling SOM domain, small activation of these two pathways are not sufficient to cause modest increase in the IP3 receptor because of the unavailability of the pKa in the microdomain. So next, we wanted to know what happens to the NFAT translocation with this synergistic stimulus. So basically, we express NFAT1 in the Stim2 knock-in cells, and we use synergistic condition. Under these synergistic condition, it causing substantial increase in the NFAT1 translocation to the nucleus. We have not yet quantified statistically, but based on the visual data, what we see in the ongoing experiment that suggests more than 60% cells showing substantial translocation in the nucleus. And when we knock down the ACAP79, it causing substantial reduction in the translocation of the NFAT1. So this suggests that synergistic condition is likely a mechanism cells use under the physiological condition, because such a small stimulus of activating both cyclic AMP pathway and IP3 signaling are sufficient to drive NFAT1 translocation to the nucleus. While in parallel, it also suggests that ACAP79 mediated scaffold is also playing a very important role in NFAT translocation. So based on our work and work by other group, we try to explain the synergistic involvement of these two pathways. So basically, ACAP, as I mentioned, that ACAP79 is a bidirectional scaffold, which one way it is scaffold pKa and regulate IP3 receptor potentiation, and on the other way it can scaffold SOC component and a downstream component which regulate NFAT. So that suggests that once calcium enters in through this SOC channel, causing increase in the microdomain, which leads to the activation of calmodulin and calcineurin eventually, and this calcineurin can phosphorylate NFAT, and the dephosphorylated NFAT then translocate to the nucleus, where it can regulate gene expression for the downstream cell function. So based on these our data, I conclude that cyclic AMP modulate IP3 receptor function at low and ambient level of IP3 receptor in the cyst cell. This regulation underlies synergistic effect of cyclic AMP at low concentration on ER calcium release, and also cyclic AMP carbachol synergistic increases Stim2 clustering, causing enhancement of the SOC and NFAT activation. And functional interaction between Stim2 and IP3 receptors enables enhancement of SOC and NFAT activation at synergistic stimulus. And finally, we also know that ACAP79 is playing a central scaffolding role in the overall in the signaling microdomain. And finally, I would like to acknowledge who have contributed in this work. So Wei Ling Ong and Sasi Rekha is helping me in the ongoing work, but also contributed in the previous work, which has already been published. Hassan Saadi, Gayon, Zehra, and Lara from David Yule Labs have contributed. And I would like to acknowledge NIDCR cell sorting facility. Basically, they helped me sorting the knock-in cell lines. And we have a very good, friendly and collaborative environment in the lab, and all this because of Dr. Ambudkar, and she's a great mentor indeed. And every day I learn something interesting from her, and I would like to thank at this occasion. We have a great collaborators, David Yule and Muhammad Tribak. Their inputs are always very useful in driving this project, and their resources, they provided us cell lines that are very useful in expediting this research, and we that lead to a conclusion in a short time. So I would like to thank David and Tribak for this, and here I would like to thank all of you to joining the talk, and thank you very much.
Thanks, Moaz. Thanks, Indu. Great talks. Yeah, we have some questions, and let me just kind of kick off. Do you mind actually stopping sharing, Moaz, if you can? Yeah, that'd be great. Let me just just start the first question. You know, in your model, like Indu, and to some extent in Moaz's model at the end, you have like this idea that Stim2 synthesizes ORI and therefore clusters stims under low level of agonist conditions. So do you perceive that at the low level of agonists that it's only Stim2 that is involved and not Stim1? Is that how you view it?
So, Moaz, I'll take that question if you don't mind.
So, yeah, please go ahead.
So, in fact, it is quite interesting, Mo, because initially, so there are a couple of ways this can happen. We don't have data yet. We just sort of started thinking about it. So if you remember Krishna's paper, it was like, the ER calcium stores are full. Stim2 is sort of partially opened. It can recruit Stim1. And the CC-CC interaction might cause opening of Stim1. So that was a model that we had predicted based on Krishna's work, the Cell Reports paper. And based on that, we said that's a possible mechanism how Stim1 could be activated when ER calcium levels are high. Now, based on Moaz's work, and we don't have any data, so this is purely hypothetical, it could be that the Stim2 plays a role in bringing Stim1 close to the site of ER calcium depletion because it's right there near the IP3 receptor. Collins also published that Stim1 can come very close to the IP3 receptor. So maybe binding to the Stim2 puts Stim1 into the right spot, and depending on how much extent of depletion there is, Stim1 could be responding to the local ER calcium change as well. But it's going to be a tough job trying to figure that out. Did that make sense?
Yeah.
Yeah.
So there are some questions here in the chat. Maybe the first question is from Don Gill. Maybe, Don, would you like to ask your question? Probably it's easier because...
Sure. Thank you, Indu. Yeah, thank you, Mo. It's a very nice pair of talks. So the question is to both of you. So I wasn't sure if the Venus Stim2 cells. I wondered, did you knock out completely the Stim1 just to see what happens to Stim2 when there is no Stim1 there? Maybe you had that. I couldn't remember.
So no, I didn't show the data, but Moaz has done those experiments. I don't know. Did we put it in the paper, Moaz? I don't know. No. But if we knock it out, if we use siRNA and knock out reduce the level of expression of Stim1, it doesn't affect the Stim2 much at all. Stim2 is still clustering.
Okay, but so I'm thinking about knockout, because if you knock down, you might still have about 10% Stim1 there, and the level of Stim1 is a lot higher than Stim2 usually. So I think
got your point. Moaz, can you answer that?
I can, yes. So basically, yes, Indu, I'm wrong. Like we have already presented that in our publication that is in the supplementary file. When we knock down the Stim1 in the knock-in cell line, basically there is no effect on the Stim2 preclusters, and it is responding well with the low agonist and high agonist. And when we knock down the Stim2 in the knock-in cell line, and we express TK Stim1 in that, basically TK Stim1 is not able to cluster with low agonist when there is no Stim2, but it cluster at high agonist. That is what
okay.
I had to do.
But so you did not actually do Stim1 knockout. You just did Stim1 knockdown. Is that right?
I did knock down both either way, but I have not used knockout lines.
Okay, I just was checking that, and I guess you don't have the MV Stim1 yet, or is that something you have looked at?
Say again.
The Venus coupled Stim1.
So we have that cell line, but basically we have not characterized that cell line. We are still doing that. But I will say, like, I have generated knock-in Estimon lines.
Okay, thank you.
Okay, Rajesh, are you there? I'm not sure if your question overlapped with that of Don, but I'll let you ask it anyway. Are you there?
Hi, yeah.
Hey, Rajesh.
Hello. Thank you for making me look cool, Indu. Yes, it kind of overlapped, Mo, with what Don asked, but I'm wondering if, if the way to go would be to look at the endogenously tagged STEM1 like you did for STEM2, to take the KI for both and to really be able to rule out if what Moaz said, if these are just tracking STEM1s passing by, because the TK STEM1, I would expect that the expression levels are still much higher than what you would have with the endogenously tagged STEM1. So I wonder if you could get your answer by endogenously tagged STEM1 and STEM2 at the same time to really be able to rule that out.
Yeah.
Yes, you're right. Probably that will address this question. Yes. So for that, we have to make a double knock-in line, basically.
Right. And then the second thing I wondered, because it appeared that the TK S2N S1C, the chimera with the STIM1 N-term domain, it looked like that you still have pre-clusters with that swap construct. So I wonder if you're suggesting that the endogenous STIM2 might also be in a partially activated state, like what we think about the overexpressed STIM2, for example.
It could be. So we didn't treat the cells with siRNA to knock down. So we can't rule out some effects of that. So yeah, good point.
Thank you.
Okay, Sandip, you around?
Yeah, hi.
Hey.
Hello.
Hi.
Good stuff. Good to see you. Really interesting. I'm just wondering if I understood you correctly. You must be having quite substantial local depletion of ER calcium in order for STIM2 to cluster. So I don't know how local that is, but can you see it? And how would you maintain local depletion?
So I don't know that. Let me answer the first one first. I don't know that you maintain the local depletion. I have a feeling at these low-level agonists, you remember there's oscillations going on. So these are probably transient things that are happening. So the question would be, once STIM2 is scaffolded to the plasma membrane lipid, how fast that can decluster. So we don't know the off rate of STIM2, like once it clusters, can it come back? I mean, once it forms these stable clusters, how soon does it come off? So in terms of the ER, so we try to look at the entire field under the turf, and you can see the decrease at the higher you go with. But then without having the STIM2 stable clusters to sort of guide us as to where we are looking for decreases, it was very difficult to pinpoint that, yeah, this decrease is real and that's not real, because it's very transient. So the only way we could sort of focus in on, we decided to focus in on what we thought were the hot spots, right? Cool spots, rather. So we use the STIM2 puncta to guide us to the ER, which is right next to that.
Right.
Yeah. So I don't know if that answers your question. Kind of.
Yeah. I mean, it would predict that there is a local cool spot, as you say, even under basal non-stimulated conditions. I just wondered if that was actually
that's because of the turnover, basal
Thanks.
levels of turnover of IP3 and any other metabolite that could affect IP3 receptors. I mean, we just talked about cyclic AMP, but that's not the only thing affecting IP3 receptors. There could be a lot of other stuff as well. Yeah.
Okay, Martin, your turn.
Thank you.
Yeah. Hey, folks. Can you hear me okay?
Yep.
Cool. Really interesting presentation. So my question was just around the differential functions of STIM1 and STIM2 in terms of the potential downstream targets of the calcium influx that they mediate. And looking at the model in your paper, it seemed to me that STIM2 and STIM1 were kind of like gears on a car. When you have low level of stimulation, you're going to basically principally have calcium influx through STIM2 mediated signaling. And as you want to go faster, and you showed this with the calcium tracers you had, you then need Stim1 to come into the puncture and to generate a lot more calcium influx, and that's where you can, if you don't have Stim1, you really affect the oscillations and things. So because they come into effectively the same puncture, the distinction between them functionally is only on the basis of cell stimulation in which they recruited. But because they're spatially localized, they're actually going to talk to exactly the same targets. It's just based on recruitment. So is in your mind, does that match everything that you've looked at in terms of targets? Are there also clusters that are quite discrete as well, where you could suggest that different things might be talked to by the calcium influx that's coming in through largely Stim2 or Stim1, or really is it just like going up, the gears in a car and you're just speeding up the influx as you recruit more Stim1 into Stim2 puncture, but they're effectively in the same place?
I mean, I think those are really, really important and interesting points you bring up, Martin, and I wish we had a clear answer, because so at these very low level stimuli, and some of us believe that would be the true, if for some tissues at least, those are the true physiological stimuli. You're not working at 100 micromolar carbachol. You're working at these very low level stimulations. And then we are sort of tuning the cells with different levels of carbachol to see, what moves. And of course, more and more depletion you get a lot of these Stim1s coming in. And then we've got all these artifacts of overexpression. So we have these big clusters. So physiologically, what are these clusters, right? We have to really go down to the endogenous levels of these proteins to really figure out what's going on. So I know that was a bit scattered of a response, but so I believe, so I think at these very low level clusters, low level stimuli, it's probably all three proteins acting together. I mean, both stims acting together. And in fact, you could have stim, there's another stim as well. There are two Stim2 proteins now. So who really knows which cluster does what, right? But the higher we go, and we can completely knock out the Stim2 and still get complete function with at least calcium influx with ORI and Stim1. So at 100 micromolar carbachol, for example, calcium entry per se is unaffected by knocking down Stim2. It's still like fully coupled, ORI is activated. So I am assuming that if you have cells where it's working at these very, very high stimulus levels, then Stim2 might not play a very important role when you reach those really high concentrations. But it may play a very important role at the lower end for tuning things and making sure everything comes to the right place. Because if it's not in the correct domain, you're not going to be able to get NF80 activation. It might not be able to talk to ion channels that it needs to talk to. So I don't know. That was a rambling reply to sort of
Indu, just to kind of clarify,
yeah,
and maybe because this Martin questions is basically the same. It's the same are all the questions that have been asked so far.
Yeah,
it's the interplay between the two stims, and
yeah.
So what you said about the involvements that Stim2 are high agonists, it is still contributing some to that current. As
Yes.
you know,
Yes.
when you use Tapsigargan and stuff, you still have, depending on the cell type, 10 to 15%.
Yes.
And so our model, which you can disagree with it if you feel like but what we suggested based on the knockouts and the quadruple knockouts and things like that is that the two stims are involved all the time. All the time. It's just the contribution. So it goes back to the stoichiometry, and so and how many do they work as heterodimers or whatever.
Right.
But the way I perceive it is that the more the concentration increases, the more perhaps you have more Stim1 in the complex than Stim2, and then the other thing happens in the lower end.
Right.
But to get to the bottom of this, maybe this double knock-ins and some more sophisticated experiments are needed. So
yeah, I think I'm absolutely in agreement with you, Mo.
Okay.
Yeah.
Can I just ask? Do you ever find lots of Stim2 puncture and lots of Stim1 puncture? I think that's. I mean, did you see the evidence for really discrete function that's spatially localized? I don't think I know that personally.
No, meaning that they are like when you co-express STIM1 and STIM2, do you actually find clusters that have either one or the other?
Yeah, absolutely, totally discrete that are activated. So there's a spatially discrete function, or are we always talking about a mixture?
So the problem with that, I think, is if we manage to do those double cells which express both endogenously tagged versions that sometime back somebody alluded to that. So I think if we have in one cell a knock-in like a STIM2 tagged and an endogenously tagged STIM1, we will be able to address it better. Once we are talking about overexpressions, I mean, using these TK promoters have helped a lot because the expression, we are tuned down the level so that we get within a twofold, higher expression using the TK promoter. But it is still much more than the endogenous. So, yeah, I think we need more work.
I was just going to say that in the resting condition, then you have, in answer to Martin's question, do you ever have one or the other in clusters? At the resting conditions, you have only STIM2 in those junctions. Is that right?
Well, if we do co-IPs, and this has to go back to our original co-IPs, and you look at endogenous co-IP data, there's always maybe, some little, so we have, I think I have to say that rather than do wishy-washy, maybe this, maybe that, I think what you make is a really good point, Don. And I just realized that we didn't do the experiment that would answer your question. So we have to sort of do some knockout cell lines and then check as to what the basal is really there.
Yeah, and having them both tagged, if you can tag them differentially, then we can answer the question about whether the endogenous Stim proteins are predominantly in heterodimers or predominantly in homodimers. That's really interesting as well, because we really don't know that. So
we don't know that. Yeah. And when Krishna did that paper that we published in Cell Reports, we were trying to get at that, basal. So we do know that if you don't express the ORI1, but you express both STIM2 and STIM1, you will pull down all three proteins right from basal state, And then when you stimulate it, more STIM1 comes in. But STIM1 is already recruited. Once you start overexpressing it, it gets recruited into these puncta, which is why this TK promoter is a great tool. You can really tune down the levels of expression to make it as close to, rather than being 50-fold above basal, we are now looking at proteins which are 2-fold above basal,
It is safe to say that under those low resting conditions, it's mostly STIM2.
Yes.
I mean, if there's some STIM1 or zero, I don't really know. So, right?
I mean, I would say that it's mostly STIM2. Yeah.
Okay. Next was Nick Dumorex. He has a very interesting question. Nick, you wanna ask it? Nick, are you there?
Yes, sure, I'm there. Sorry.
Hey, Nick.
Hey, thank you for the presentation. Nice to see you all, guys. Just had a question about the molecular crowding in these junctions, because we keep putting more adapter proteins, scaffolding proteins. The ACAP, as far as I know, is quite a big complex, like 17 nanometer in length. So I wonder how that would fit in the junction without disturbing the productive interaction between the Stim and OI.
Good question. I don't have an idea, actually. Maybe it's something I need to think about. I would defer to more of the people who've been looking at what are the structural details.
I'm not 100% sure, but I think the STIM2 is a bit longer than STIM1, so would that maybe be favorable for the STIM2, interacting at larger gaps compared to the STIM1?
I wish I had an answer to that, but I think you raise a very, very important point there. How many things can you cram into those junctions? I think we had sort of discussed it, and I don't know whether. I wish I knew. I don't know.
And the other point, you showed nice co-localization of the mapper and the STIM2 in the clusters. But we see that also. But when we co-express the ORI, and then you see the STIM and ORI cluster, the mapper is pushed away. And we interpret that as, again, a question of gap distance, you know. Did you ever try that?
Yes, Indu, yes, not tried, but basically we have not included in the publication, but when we express mapper, we see that it's not like on the top, ORI one and mapper not on the top of each other. They're localizing somewhere like juxtaposed.
Okay.
But we need to, but I need to do more experiment to make any conclusion on that.
Oh, we did those? I don't think.
Yes, we have.
Okay, thank you.
I was just going to comment on, so Nick raises an interesting question. STIM2 is longer in the C-terminus, but most of that, all of that is in the flexible domain. And so the distance between the ER membrane and the top of SOR in STIM2, which is probably the distance that across the junction, would probably be the same for STIM1 and STIM2.
Mm-hmm. Nice point, yeah.
Okay, well, I guess there are no more questions. This was great discussion.
Yeah.
Cool stuff, Indu and Moaz.
Always nice to see everyone.
Yeah, it was good to see everybody. So until next one, Nick, not Nick, sorry, Martin, do I hand it to you? I'm not sure if you want to do like you do in the past. Keep the conversation open, or just
I don't mind. I mean, if people want to stay and chat, the room can stay open. It's absolutely fine. But I just want to thank everybody who came along. I also want to thank Katja, who and Marlene, of course, and also Adelina Ivanova has joined us in organizing the webinars too. So thank you very much to them, and thanks to everyone for coming along. We're going to do these once a month, so we'll see you about the same time next month. If you can come along, please do. Please drag your lab along as well. If anyone wants to stay and chat, they're most welcome.